Lecture 7. Causative agents of bacterial respiratory infection, pathogenic mycobacteria. Causative agent of tuberculosis. Okay, classification. The causative agent of tuberculosis is belong to order Actinomycetales, family Mycobacteriaceae, genus Mycobacteria. To date, more than 130 Mycobacteria species are known, but pathogenous are Mycobacterium tuberculosis is a predominant causative agent of tuberculosis in humans, and Mycobacterium bovis causes tuberculosis in cattle and much more seldom in humans. Mycobacterium africanum can rarely produce human tuberculosis infection, and Mycobacterium avium, intercellularum, Kansasi, ulcerans, and others, more than 60 species, are causative agents of mycobacteriosis. They usually affect immune-compromised individuals, for example, AIDS patients. And Mycobacterium leprae is a causative agent of leprosy. So you should notice that there are a lot of species in the genus Mycobacteria, but the most spreaded is Mycobacterium tuberculosis, causative agent of tuberculosis, and also pathogenic is, but not so spreaded, are Mycobacterium leprae, and all others are conditionally pathogenic, and one group is the pathogens of mycobacteriosis. Mycobacteria are thin strain rods. They don't have capsules, spore, and flagella. They are highly polymorphic, and bacteria can appear in granular, coccoid, and thread-like branching and filtering form, so very different shape. They are gram-positive but poorly stained with aniline dyes, so they don't. They have gram-positive cell wall, but it is not easy to stain them with gram staining. That's why more comfortable method of staining is Ziel-Nielsen staining. Striking acid and alkali resistance, so-called acid fastness, these abilities ensure from particular chemical composition of Mycobacterium. They contain the great number of chemical inert lipids, phosphatides, and waxes, usually termed as mycolic acids, various high molecular weight hydroxyl fatty acids in a complex with cell wall. Mucopeptides, besides lipids, fraction, bacteria include various proteins and polysaccharides. Their cell wall um, uh, is consists of a lot of lipids and waxes, and they made mycobacterial cell wall non-sensitive for acid and alkaline action. So they are resistant for alkaline and acid because of lipids and waxes in their cell wall. And the stain, red basic phenol fuchsin in Ziel-Nielsen stain, whereas other bacteria are sensitive to sulfuric acid treatment and contour stain with methylene blue. Also, mycobacteria are successfully stained by fluorescent dyes, for example, auramine. Usually use Ziel-Nielsen stain, and they are red. They are colored in red by this method of staining. Okay, here you can see Ziel-Nielsen staining and thin, long rods. red because they are stained with Zell-Nielsen stain. They are acid-resistant bacteria. The cell wall consists a lot of additional lipids, glycolic acids, and waxes that made cell wall resistant for environments factor for phagocytosis and acids and alkaline. Here you can see glycolic acids, arabinogalactan, and other substance. Cultivation, they grow very slowly in aerobic condition. It depends on long period of microbial replication, about 15 hours for doubling. And they take about 1 month to make the first colony. So the cultivation is very slowly processed. Temperature range from 24 to 42 degrees with optimum at 37. Lowenstein Jensen medium contains agar, egg yolk, glycerol, potato extract, asparagine, milk, salt, and malachite green to inhibit contaminate flora microbiota. Primary growth on solid media is observed in 3-6 weeks, very slowly. Mycobacteria tuberculosis usually grows in warty dry colonies, air forms with cream-colored ivory pigment. Saprophytic mycobacteria proliferate more rapidly and appear in a few days. They are able to produce orange or yellow pigment. Different growth bacteria support the cultivation of small amounts of mycobacteria. Liquid media growth reveals thin, brittle, fringlet film result from microbial hydrophobic substance. Here you can see Levenstein Jensen media and our colonies with ivory pigment formed by Mycobacterium tuberculosis. And also they are biochemical properties. Mycobacterium tuberculosis are aerobic. They produce number of oxidation reduction enzymes, including thermolabile catalase peroxidase and superoxide dismutase, produce niacin, express lecitinase, phosphatase, and urease, can utilize various carbohydrates and proteins. Biochemical properties, Mycobacterium tuberculosis are, antigenic structure. They have lipid antigen and lipid and phosphatides encased in mycobacteria are generally regarded as a moderate antigens or haptens. Nevertheless, in complex with mycobacterial proteins, they elicit both cellular and humoral immune response. Mycobacterium tuberculosis infection induced cell-mediated reaction of delayed type hypersensitivity. Delayed type of hypersensitivity with chronic inflammation, with hyperactivation of macrophage and cellular inflammation. Also, mycobacterial antigen activate production of specific antibodies, usually in low moderate titers. Tuberculin is a peculiar antigen complex composed of various tubercular proteins and wax fraction. It causes hypersensitivity reaction evaluation by tuberculin skin test (TST). Tuberculin is associated with delayed type hypersensitivity. So it's an allergen which causes delayed type of hypersensitivity. And TST test, it's reveal the presence of infectious allergic reaction. Virulence factor, toxic substance of mycobacteria are closely associated with microbial body and mainly released after microbial decomposition. So factor, factors of violence are present in the cell wall. All these lipids, acids, waxes are the main violence factor, and the main its target it's a phagocytic cell to broke phagocytosis. So mycolic acids render toxic effect against host cell and tissue. Cell wall structure of glycolipid nature, mycozids, monozids, etc. Participate in microbial adhesion and inhibit phagocytosis. Mycobacterium tuberculosis possess highly specific type 7 secretion system that promotes active secretion of Mycobacteria proteins across lipid-containing cell wall of Mycobacteria. T7SS is characteristic for violent strains of Mycobacterium tuberculosis and Mycobacterium bovis, being absent in vaccine B GG strain with the 8T7SS. Mycobacterium tuberculosis expels some virulence affected proteins into the cytoplasm infected cell, like macrophage. Among them are CFP10 and SIAT6, which inhibit respiratory burst and secretion of pro-inflammatory cytokines by macrophage. Other proteins prevent the recognition of mycobacteria by toll-like reception of immune cell. So all these factors break down phagocytosis by breaking down hemotaxis and respiratory burst and the process of recognition and other. So it's non-effective phagocytosis. It's a uh, main part of the tuberculosis pathogenicity. Microbial enzymes catalyze and superoxide dismutase contribute to inhibitory of respiratory burst by macrophage. Mycobacterium tuberculosis carries so-called court factor for potent toxic glycolipid fraction, which inhibit biological oxidation in the host cell and induce chronic immune inflammatory response with granuloma formation. Tuberculin is toxic for genopeaks in course of experimental infection and elicits hypersensitivity in human infection, followed by tissue inflammation. Tuberculin cause hypersensitivity, delayed hypersensitive reaction. A mutation in gene encoding bacteria enzyme catalyze and RNA polymerase confirm the resistance of mycobacteria to basic tuberculosis drugs of the first line isoniazid and rifampicin. Resistance, Mycobacteria tuberculosis, they have very high resistance because they have very resistant cell wall, and they remain viable in water for 1 year, in soil for 6 months, in sputum several months, in dried sputum. Due to higher lipids content, the bacteria can withstand with the action of generally used disinfectants. Thus, ordinary disinfection regimen should be prolonged to inactivate mycobacteria. The most efficient are chlorine-containing chemical agents. Mycobacteria are resistant to majority of microbial drugs. Heating at temperature of 100 degrees readily kills all mycobacteria. So it's a very effective method to kill bacteria, to kill mycobacteria. Pasteurization inactivate mycobacteria, bovis dairy products, thereby preventing alimentary transmission of the disease. Mycobacteria are susceptible to sunlight and ultraviolet irradiation. but they don't kill very quickly. It takes some time. Epidemiology. Tuberculosis is one of the most important traits of the human health at the beginning of the 21st century. According to WHO data, annual total number of diseases cases is about 9.6 million. It is general assumes that about one quarter of affected person dies from tuberculosis and its consequence, and most of patients are young adults. Mortality rate in untreated or untreatable disease exceeds 50%. Untreatable disease means that antibiotics are not effective. That's why this disease is untreatable. Yes, it is very big problem. The ineffective ineffectiveness of antibiotics in some cases of tuberculosis. Um, Tuberculosis is a major AIDS indicator disease in HIV infection person, and the rapid spread of multi-drug resistant MDR and extensively drug-resistant exterior tuberculosis. Now, about 3.5% of tuberculosis cases worldwide are produced by media mycobacteria, and in certain world regions, African countries, several Chinese and Russia provides Baltic states, the incidence greatly exceeds. Therefore, the global spread of MDR tuberculosis is a big problem of great medical and social importance. In our region, in Yakutia, we have a lot of cases of tuberculosis, and it is about extremal climate condition and extremal low temperature in our winter. That's why respiratory infection are very spreading in our region, and especially tuberculosis. Tuberculosis remains the social disease that strongly correlates with poverty, adverse living and working condition, and general economic decline. The main source of infection are persons with active tuberculosis and rarely sick animals, cattle. The infection is transmitted predominantly by airborne, air droplet route, and more seldom by contact route. Ingestion of contaminated foodstuff, usually milk, is also possible, especially for Mycobacterium bovis infection. Very seldom the disease can be transmitted by direct inoculations that may occur among healthcare workers. Overcrowded, malnutrition, and starvation, insensibility of medical care, and other hard socioeconomic conditions, as well as separation of patients' immune system, are in the direct relationships with tuberculosis susceptibility and pathogen dissemination. Pathogenesis and clinical findings. After primary penetration, Mycobacterium infection usually remains latent. Without prophylaxis, about 5 or 10% of infected persons produce tuberculosis disease. Inhaled Mycobacteria are ingested by lung macrophage and transported to the regional bronchial lymph nodes. So, lymph nodes is the first who take Mycobacteria. So, macrophage, they can't digest, they can destroy bacteria, and bacteria goes on and the bacteria can multiply inside the macrophage and goes inside the micros and goes to the lymphoid nodes by macrophage. Um, it is because of ability of Mycobacterium tuberculosis to block phagolysosome and to break down phagocytosis. Court factor inhibits cell migration with the inflammatory focus. So Mycobacterial tuberculosis survives in phagocytes, blocking phagolysosome fusion. Here you can see that bacteria present in the phagosome and don't and break down the phagolysosome fusion, and also it can neutralize lysosomal enzymes, and it can multiply inside the macrophage. So in the case of um, um, virulent state Mycobacterium tuberculosis, it caused the process of uh, infection of macrophage and destroy it, and then infect other macrophage. If you have attenuated Mycobacterium tuberculosis, so after vaccine and macrophage, um, uh, they cause apoptosis of infected macrophage, restriction of bacterial replication, and phagocytic apoptotic vessels. They goes to the interaction with T cells and interaction with CD8 cell uh, and activation of adaptive immunity. Primary lung tuberculosis is characterized by acute exudative lesions affecting lung axillary tissue with subsequent rapid involvement of lymphatic vessels and regional bronchial lymph nodes. Primary tubercular complex cell-mediated inflammation leads to formation of tubercular granulomas, tubercles with caseous necrosis in their centers. Multinucleated giant cell, epithelioid cell, lymphocytes, macrophage, and fibroblasts surround inflammation focused with mycobacteria. Typical symptoms of progressive pulmonary disease include intoxication, fever, productive coughs with hemoptysis and enlargement of lymph nodes and abnormal results of chest X-ray examination. So, um, okay. This is granulomatous tubercles. It's they forms because of delayed hypersensitivity reaction. This delayed hypersensitivity is caused by tuberculin, the protein of cell wall of Mycobacterium tuberculosis, and Mycobacterium tuberculosis. Uh, they after activation of T cell and T helpers activated uh, because of interaction with the dendritic cell. Dendritic cells, they or other antigen presenting cell, they make a sensibilization of T cell, CD8 cell, and also they activate macrophage, and in the place where tuberculin allergen bacteria presence, uh, all a lot of cell migrate. So here you can see which cell presence in granulomas. It's epithelioid macrophage, a lot of, and neutrophil, T cells, natural killers, also dendritic cells. So the light hypersensitivity means that bacterial tuberculin is allergen and After process of sensibilization, uh, different immune cell they infiltrate the place of presence of uh, allergen, the place of uh, contain the place which contain Mycobacterium tuberculosis, and this infiltration is called tuberculogranulomas infiltration with immune cell. In the center of this infiltration is a place of necrosis, and this granulomas it can be encapsulated so by uh, make of make fibrosis or in the on the other side it um, can go to the uh, to it can become bigger, and the zone of necrosis can be bigger and bigger and bigger, and this is a worse condition. Here you can see histology. Histology picture of uh, uh, tuberculosis granulome. So here you can see the zone of necrosis and the zone of different immune competent cell. Cellular immune reaction restricts inflammation area and terminate the propagation of Mycobacterium tuberculosis. Primary specific process is resolved with connective tissue progression, fibrosis, and calcification. So here we can see the granulomas, and if immune system can encapsulate it, can make a fibrosis and calcification. These granulomas became dormant state, non-active states, non-active state. But inside this dormant state, live bacteria presence in this uh, dormant state, and if and this dormant state they kept all the patient's life, and if this patient have a strong immunity, it will be everything okay. But if the immunity became lower, this dormant state can reactivate and make a secondary tuberculosis. Yeah, it's how to spread. For spreading, it is necessary to have a conversation, to have a entering of mycobacteria from the granuloma to the bronchial tree. If the granuloma is not connected with bronchial tree, this patient will not be uh, will not be uh, will not be contagious. In the case of decreased immune resistance, especially combined with a large microbial load, further spread of pathogen is possible via bronchi, lymphatic, and blood vessels. Disseminated disease affects lungs, miliary tuberculosis, or leads to extrapulmonary tuberculosis, tuberculosis of eyes, intestine, kidney, tubercular meningitis. These forms are much more severe and may cause patient death. Reactivation of viable mycobacteria is possible due to many unfavorable external or internal stimuli and easily affects immune compromised person. Secondary tuberculosis is characterized by chronic tissue lesions, followed by disseminated fibrosis. Secondary lesions are very difficult and treatment showing no tendency to self-recovery. Okay, so this is a form of tuberculosis. Immunity. Immunity is cell-mediated and non-sterile, maintained by viable mycobacteria. Non-sterile means that immunity presents only while bacteria presence in the organism. Macrophage, dendritic cell, and T helper cell subsets produce a vast number of pro-inflammatory cytokines, thereby inhibiting microbial propagation. Antibodies are not proven to possess substantial antimicrobial activity. As it was mentioned above, human population has high natural resistance to Mycobacterium tuberculosis. Laboratory diagnosis. So there are a few methods: bacteriological, microscopical, molecular PCR. And for bacteriological methods, the specimens can be taken from sputum, lymph nodes, puncture contents, urine, and cerebrospinal fluid if it is disseminated tuberculosis. Microscopy of acid phase bacilli is available primary test for laboratory diagnosis of the disease. Tuberculosis bacilli strain read by Fuchsin in Nielsen method due to the remarkable acid resistance of bacteria. So fast and primary test is staining of sputum with Nielsen stain. And fluorescent microscopy also very sensitive and can be applied. Cultivation is not very rapid, it's very slowly, and that's why bacteriological method of diagnosis can be applied only for detection of antibiotic resistance, only for establish the sensitivity of antibiotics and monitoring of treatment. So you should know that bacteriological method is not good for diagnosis, but it's one and main method to control the treatment, to find the best antibiotics um, for this bacteria for each cases. And also there is a price microcultural method, and before PCR we used price method to rapidly diagnosis. But now we have PCR, and so it is the best method for rapid diagnosis of tuberculosis. And also we have tuberculin skin test, TST, or MAN2 reaction. Its evaluation delayed hypersensitivity in Mycobacterium tuberculosis infection. Tuberculin is a multi-component antigen complex of Mycobacterium tuberculosis composed of various tubercular proteins and FACS fraction. Robert Koch obtained it's a glycerol-based filtrated suspension of killed tubercle bacilli. He applied it for tuberculosis treatment but without evident success. Nevertheless, tuberculin was proven to be worthy for diagnosis of tuberculosis. It was further purified to derive protein fraction, tuberculin PPD, after intracutaneous injection of a defined amount of tuberculin to the patient previously exposed to Mycobacterium tuberculosis. The paper will appear begin maximum in 24-72 hours. A positive TST indicates that the person has been infected with Mycobacterium tuberculosis. The explanation may be difficult because previous BCG vaccination, specific chemotherapy, or host immune status can influence on the reaction. However, test conversion from negative to positive implies recent infection and possible current activity of tuberculosis. A positive skin test assist positive skin test in assisting diagnosis, and it's also helpful for evaluation of tuberculosis treatment. Here you can see tuberculosis skin test, and tuberculosis skin test it's revealing the uh, hypersensitivity or the light type of hypersensitivity. So tuberculin it causes hypersensitivity, and when it injected in the skin, the process of cellular inflammation in the skin is began. Antigen is injected into subcutaneous tissue and processed by local antigen presenting cell, and Th1 effector cell recognize antigen and release cytokines which acts on vascular endothelium. Recruitment of phagocytes and plasma to site antigen in injection cause visible lesion. Visible lesion is because of inflammation caused by cell reaction, which is characterized hypersensitivity type 6. So, TST detects T cell response to Mycobacterium tuberculosis antigen, exactly to tuberculin. And more advanced version of allergic skin reaction is tuberculosis is recently devised the skin test DST. It uses recombinant protein of Mycobacterium tuberculosis as infection mycobacterial allergens for intracutaneous injection. This test has serious advantages against TST, and it is not influenced by primary BCG vaccination. BCG bacilli are lack of CFP10 and ESET6 virulence proteins. So, uh, if we use TST, all BCG vaccinated person will show the reaction, and if somebody have its individual higher reactivity, the TST test can be false positive. But DST test it's more sensitive because BCG vaccinated person must not react to this test because it consists only proteins which are not present in the cell wall of vaccine strains. So vaccination is not um, uh, influence is not influence on the reaction, on the result of reaction. That's why nowadays, if if the person uh, have the incidence of allergy, it's more comfortable to use dial skin test, not TST test. Treatment Course of tuberculosis patients lasts for 6 to 12 months. Long duration of treatment period ensure from slow metabolism of tubercle bacilli as they tend to intracellular persistency and permanent invasion from host immune system. And antibiotics of choice are isoniazid, rifampicin, pyrazinamide, ethambutol, and streptomycin. And pyrazinamide is a sole antibacterial drug that can affect intracellular forms of Mycobacterium tuberculosis. Combined use of first-line drugs for 6 months of so-called short chemotherapy course, recommended by WHO, yields cure rates raises at 80-90% and prevents the emergence of drug resistance. Second line preparation, fluoroquinolones, ethionamide, cycloserine demonstrate lower efficiency but often increase toxicity being of more seldom use. And the treatment of MDR and XDR tuberculosis poses serious difficulties. AMDR bacteria are resistant to isoniazid and rifampicin, whereas XDR microbial cells are additionally resistant to fluoroquinolones and one more drugs of the second line. So it is very difficult to treat this infection which caused resistant strains. Prophylaxis. Non-specific prophylaxis of the disease is achieved by isolation and adequate treatment of tuberculosis patients. And vaccination, it's a specific prophylaxis and it's made with attenuated BCG vaccine. And this vaccine is contained of a virulent attenuated strain of Mycobacteria bovis obtained by Calmet and Giren. And newborn infants undergo primary vaccination at 3-5 days of life. Human immunization with BCG vaccine reduces the risk of tuberculosis in vaccinated person by about 50%. Okay. And also this vaccination prevents several form and hard form of infection. Thank you for attention. If you have any question, you can ask me in WhatsApp or my email.